Hello, my name is Alfredo, and today I'll be talking about the disease anthrax. This infectious disease occurs from the bacteria Bacillus anthracis, and its common names include malignant edema and Wool-Sorter's disease, yet its formal name is just anthrax. In regards to the taxonomy of B. anthracis, it belongs to the domain Bacteria, the phylum Firmicutes, the class Bacilli, the order Bacillassus, the family Bacilliaceae, the genus Bacillus, and there's a subdivision of the genus being the group Bacillus cereus, and its species is Anthracis. The members of the B. cereus group are very similar to each other in terms of their phenotypic characteristics and their host preference. For example, B. cereus, the species within the B. cereus group, is able to cause many diseases such as bacteremia or septicemia, endocarditis, and meningitis, as well as 2 food poisoning syndromes. B. anthracis is a gram-positive, rod-shaped bacterium with a size of about 1 micrometers by 4 micrometers. You can see this here in the diagram on the right, where its arrangement is just rods and as opposed to spheres, and its purple color indicates that it is gram-positive. It is also found in soils all throughout the world and grows at mesophilic temperatures, and it is a facultative anaerobe. It does have one single gene for flagella, but those 4 proteins associated with it are mutated with frame shifts or point mutations, and therefore that flagella is not actually functional. The structure and replication of B. anthracis. It includes endospores that are responsible for all types of the disease. And these endospores are made of a core and a cortex, which allow for total metabolic dormancy and protein stability, making the spores very tough and able to withstand very adverse conditions. Once they are infected into the host body, B. anthracis will replicate when the spores germinate and then become vegetative cells. When the host cell dies, the vegetative cells will return to the spore form, which are highly resistant and able to survive in the body for a great amount of time. As seen in the picture, the majority of the endospore is composed of the core, and the outer area is that cortex, and both of these structures allow for a very toughness and rigidity of the structure. The history of anthrax. It has existed since the ancient Egyptian times, and the Greeks even included it in some of their famous writings. In 1752, Meric came up with the first clinical descriptions of the disease, and in 1877, Robert Koch developed his famous Koch postulates. He was able to determine the life cycle of B. anthracis to show a relationship between a certain organism and a disease that correlates with each other. The pathogenesis of B. anthracis. This organism relies on its virulence factors found on 2 distinct plasmids, PX01 and PX02. Plasmid PX01 encodes for 2 different toxins, lethal factor, also known as LF, and edema factor, OF, which both have a structure called PA, a subunit that binds to receptors TEM8 and CMG2 on the cell membrane of the host cell. Once the PA has binded to the host cell, the LF and EF are transported into the host cell. This is when they start their toxic activity when we first look at LF and what it does in the cell. LF will cleave the N-terminal of MAPKK, inducing cell lysis and EF will catalyze a conversion of ATP to CAMP, diminishing cytokine response in the cell and lowering the overall immune response. Plasmid PXO2 is responsible solely just for producing a protective capsule that surrounds the bacteria and defends it against macrophages and other defense cells within the immune system. Looking at the image here, we can see how B. anthracis produces EF and LF, and they will both migrate to the host cells, where then PA will come in and be cleaved off by a furin, which will allow for it to join either EF or LF. And once that EF-PA or LF-PA complex enters the host cell, they'll be able to start their cytotoxic activity. We can also see how B. anthracis is surrounded by that protective capsule mentioned earlier, thanks to the other plasmid, the PXO2 plasmid, and this will prevent it from being eaten by other immune cells and allowing it to survive longer in the host body. Moving on to the different types of anthrax, and there are 3 total types: the cutaneous form of anthrax, the inhalation form of anthrax, and the gastrointestinal form of anthrax. The cutaneous form of anthrax has approximately 1 to 5 days for incubation period, and it will be acquired through direct physical contact with contaminated animals. So the majority of the time this will occur for farmers or rural workers who do not work in the most sanitary environments and have to deal with animals who are already predisposed, already have been in contact with B. anthracis. And it's in their manual labor that they will directly come in contact with that organism, and then they will develop the cutaneous form of the disease. For the inhalation form of anthrax, its incubation period is anywhere between 1 and 60 days, which makes it a lot different than its other incubation periods of the other 2 types of the disease. It's acquired through breathing it in from the endospores, and the endospores being trapped into the lungs. And lastly, gastrointestinal disease comes from, or the form of anthrax comes from eating contaminated food, such as poorly cooked meat. And its incubation period is about 2 to 5 days. The symptoms and signs of anthrax. For the cutaneous form of the disease, which is also the most common type of disease of anthrax, the first symptoms include itching, and then a very large sore or boil will appear on the skin. And if it's not treated immediately or soon enough, it can easily spread to the lymph nodes or to the bloodstream, which further complicates the disease. For the symptoms of the inhalatory form of anthrax, the first ones include fatigue, fever, a cough, and then there will be severe difficulty breathing and rapid pulse between 2 to 5 days. And this form of anthrax is also the most rare in the US. And for gastrointestinal, the symptoms include fever, severe abdominal pain, bloody diarrhea, and vomiting with blood. For the treatment of anthrax, the standard procedure is antibiotics such as ciprofloxacin, doxycycline, or levofloxacin. For inhalation anthrax, intravenous therapy is most effective along with other antibiotics such as penicillin and ampicillin. There are also antitoxin therapies that have been developed for inhalation anthrax such as raxibacumab and obeltoxaximab. These antitoxins are unique in that they will target the toxins produced by the bacteria instead of actually targeting the bacteria themselves. In the prevention of anthrax, the first animal vaccine was developed by Louis Pasteur in 1881, and he injected 25 animals with his first form of the vaccine, and all 25 of them survived. Another animal vaccine was made by Max Stern in 1937, and this significantly reduced the number of human anthrax cases to the point where there was only 18 cases of inhalation anthrax in the United States. And by the 1950s, the first vaccine for humans was made from stabilizing and purifying that PA subunit that binds to LF and EF by precipitation, and it was 92.5% effective. Thank you for watching.